showed you in the previous slide with that iGenomes base. And that's pretty much it. And then when you run the pipeline, I'll show you a bit later on how to provide that. Every pipeline has to have a test data set. So this is vital for the pipeline development, because you want to be able to test your pipeline constantly whenever you push commits and you make changes to the pipeline. Also the fact that it's going to be developed by the community, so you want, anytime that a change is made to the pipeline, you want these tests to be carried out automatically so the pipeline is still functioning. So here I've got, I think it's a ChipSeq one. It's just a design file. It's a simple yeast data set that is downloading some publicly available data that's been subsampled to, I think, 100,000 or 1 million reads, which is ample for yeast. And this will then get run if you just provide profile test. So going back to the continuous integration, which I was just talking about, it's important that you make sure that you have a working pipeline at all times before you merge it into your main code base. So we use Travis CI for that. And the first thing we do is lint the pipeline to test for best practice, NF-core best practice in the template of the pipeline itself to make sure that everything should be as it is, the containers are tagged properly and so on. We also do a markdown lint for any markdown documents we have in the repository of the pipeline. And then we run a test version of the pipeline before we merge it. And this has to pass before we merge it. There's common usage. So as you can see here, the top 4 pipelines here, the input is all sort of FastQ files. The bottom 3 there is design files, which will contain paths to the FastQ files, along with additional information about the experiment, like inputs, controls, and so on. Now I will try and give you a switch screen to this. I think this might happen. Do I have to escape it? Or do I have to pull it to another side? So this is just in a web browser. Oh, I see, we've got to drag it across. Nice, okay. Sorry. So, I'm going to drag the entire window. Right, so this is me running a test version of the ChipSeq pipeline at the CRIC. So we have environment modules at the CRIC. So I'm just going to create a test directory. Just going to purge all the modules in the environment. Load Nextflow itself. And then load Singularity, because that's what I'm using on the cluster. And those are the only 2 dependencies you need, in theory, to run the pipeline. So now I'm just saying I want to run the test data set with the profile test, and in conjunction with the CRIC profile, which brings in the Singularity and all of those settings that I had. And I want to run version 1, and I want a nice email at the end of the pipeline. So if you don't provide Nextflow will download the pipeline for you automatically if it's not already there existing locally. If the container for the pipeline isn't local either, it will download that for you as well. But you can obviously also override that and give it a local container. If you're running the pipeline multiple times, you can just give it a path to a container. But in this case, it's not available. Assuming it's not available locally and you're running it for the first time, it will download it for you. So let's forward this a bit. And then the pipeline kicks off. So Nextflow will sit on your login node, for example, and it will just submit all of your jobs to your cluster, and they will just complete. I think I've got 6 samples possibly in this experiment, and it will just run through. I'm just speeding it up. Right, pipeline's finished. In terms of the directory structure, you have a results directory and a work directory. So the work directory is a sort of a standard Nextflow directory, and it essentially caches all of the temporary data and intermediate files from the processes that Nextflow carries out. And it's useful if you want to, say, for example, resume the pipeline. If it fails at some point and you want to be able to resume it, all of the information that it requires to rerun and resume from that step is in there. The results directory contains the results. So FastQC contains HTML files, FastQC. Trim galore, again, after adapter trimming, some FastQC files. There's an IGV session with paths to bigwig and peaks and stuff that are generated by the pipeline, so you can just load it up in IGV. Also, Nextflow is great in terms of the reporting, so you get a bunch of reports telling you more about the resource usage, which is great for debugging. And then you then have also your final filtered BAM files in another directory, along with your peaks, bigwig files, and a bunch of QC metrics that you've generated via the pipeline. So that's the demo. At the end of the pipeline, you can get a nice flashy email telling you that it's finished or failed. It comes attached with a multi-QC report. So this is the chipseat pipeline, so you can just scan through it quite quickly and see how your data looks straight away. There's some custom content in there that I've put in as well. It's quite nice. These are the standard reporting tools that Nextflow has. It tells you resource usage, memory usage, and so on per process, so you can help debug things if your pipeline's failing, for example, even for benchmarking. There's another example of that, which tells you how long each process took and memory and memory requirements. With each pipeline as well, documentation is key. We all know that. There's extensive documentation on the pipeline, where the files are, and some example plots to help guide the user, and, so they can read the documentation and then work out what's going on and hopefully QC and analyze their own data. There's also extensive documentation on the usage, so all of the parameters that you have for the pipeline. These are all documented here, for example. Back to the presentation, try and drag this across. We're also continually developing a number of tools to help in the creation of pipelines. So it's called NF-Core Tools. It can be installed quite easily by a pip. It allows you to create a blank slate template of a pipeline if you're a developer, for example. And this will contain the latest sort of I guess best practice and also a bunch of boilerplate code that you don't really want to have to write yourself. You can list pipelines, you can download pipelines if you're running in an offline environment. You can lint pipelines, which also is quite important to make sure that your pipeline is adhering to the best practices that we set. You can bump versions, so just before you release, you can go from a dev to a version tag, and it just essentially updates the pipeline version in all the correct places for you, so you don't have to do that. This is outdated now, but there's now sort of 22 institutions and 70 contributors on GitHub that have contributed to NF-Core. There's about 155 members on our Slack workspace, which is great. And you can communicate with us via any of these methods, create an issue if you see a problem with the pipeline, contribute, which is even better, via pull request. Some of you may have already seen our bioarchive paper, and we also have a manuscript submitted, and it's currently under revision. So some future things, we want to be able to implement sort of module syntax within our pipelines. And this is quite a recent feature that's been added to Nextflow. It's been in SnakeMake and other languages probably for a lot longer. But it's quite primitive, but it'll be great when it's there because it will strip away a lot of the syntax and it will just give people a lot more flexibility in terms of using the same modules across pipelines and so on. We've also got some future plans to get more accurate cloud computing costs, to integrate biocontainers, which again is great because then you can, per process, you can just specify an individual biocontainer and run the process in that way, and it will be fully reproducible because they're all within Docker containers. Automated testing for full-size data sets. So at the moment, as I mentioned, we're doing the testing on minimal data sets, but before we release, it'd be nice to be able to test on larger data sets just to put the pipeline through its paces, essentially, before you release. So I'd like to thank Phil Yules, Maxime Garcia, Alex Peltzer, who's talking at BOSC on Thursday, Sven Fillinger, who have helped create and maintain this incredible community, Paolo Di Tomasio, who's the creator and lead developer of Nextflow, and also Evan Floden, and the communities themselves, so the NF-Core and Nextflow communities, and my group for putting up with me for over the years. Thank you.
Great, thanks. Do we have any questions?
Is there a GUI that will enable non-programmers to use these workflows, or do you plan to develop GUIs for the workflows?
In what way? So do you mean to contribute or to run them?
Is there available GUIs for Nextflow workflows?
GUIs, so we do have these things. They're at the moment, they're open pull requests. So we're working on them.
Okay.
So command line type tool that would allow you to, or will prompt you to add in parameters and stuff, for example. We're working on that. We're also trying to work on, so this is at the pipeline level at the moment. We're also trying to work on having an overview. So if you imagine a lot of core facilities, they have a high throughput of data, different types of data. So it'd be nice to, providing that you have the right metadata, which is actually key, you can imagine the data coming off the sequencer, getting relevant information from a LIMS system or something like that, and then just automatically pushing the pipeline through to be analyzed. So we're looking at ways, hopefully, to try and monitor all of that as well. So you can submit multiple pipelines and see how they progress, whether it's failed, whether it's passed, and so on.
So I guess this question is for you, and in general, I think for a lot of the people in this room who are using pipelines of some sort. And the issue here is, yes, you can get things to go through a pipeline, and there is definitely a set of users that we all know who think that as long as a computer program has worked, the results must be right. So the onus here is basically on the users to make sure that the results are sane, or is there any effort being done on the developmental side of the pipelines themselves to decide if the results that are coming out are releasable or not?
So ideally, the developer of the pipeline would integrate steps that allow you to QC the data well enough for you to make a decision whether the experiment itself has worked. The pipeline, as you say correctly, won't make the biological interpretations for you, but I don't think any pipeline will. So it comes down to, having said that, we do have on our Slack workspace, we have pipeline channels. So if you've seen that your data doesn't look right, then you can always post on the channel and say, look, this is what my data looks like. Have you seen this before, or can you help me? But the data, the downstream data and results it's up to the scientists to work out what their data looks like and then to take it further from there. Hopefully the QC can guide you.
Any other questions? All right, great. Thank you, Harshil. I'd like to thank all of the presenters.